Our next presenter is George Merhej from Drug Discovery and Biomedical Sciences. Please welcome George.
Imagine trying to identify your enemy among a group of people who all look the same. Similar height, similar hair, similar facial structure. How do you tell who your enemy is? It's hard, isn't it? Now, if I tell you that your enemy has one special feature, they have a bigger nose, wouldn't this make it easier to tell who your enemy is? In my PhD research, I tried to identify the enemy by going after the bigger nose. Now, I don't go after people with bigger noses, that's the allergen. The enemy in my case for my research is a protein molecule called PLK1. It's present in our cells and it has roles in cancer growth, makes cancer grow. So it's the enemy, it's the bad guy. And the big nose in the case of PLK1 is a small part of it called the PBD of PLK1. That's special of PLK1, specific to PLK1. Specificity, this is a big goal in my research. Now, if you think through this a little bit, for the example I gave, if I was going after the height or the facial structure, would I be able to catch my enemy? Probably, but I would be catching the friends as well, because those are common features between the friends and the enemy. Going after the big nose, however, would get me to catch the enemy specifically. And you've probably heard before that the big problem with cancer medicines, with cancer drugs, is that they might kill cancer, but they also cause trouble to our normal organs and normal cells. I want to avoid that. So I try to develop drug molecules that bind specifically to the enemy, to the PBD of PLK1. To do that, I use a variety of AI tools and softwares. And then after developing those molecules, I make them in the lab, I produce them. Then third step would be testing them to see if they're working. Testing them in a cancer setting in the lab. What does a cancer setting look like in a biomedical lab? It's basically an animal that we get to have cancer to mimic a human with cancer. I test those molecules in that animal and I look for two main things. One, obviously, is curing cancer in the animal, so the smaller cancer tumor. Two, back to specificity. So I want to see killing of the enemy while saving the friends, which are basically the normal organs and normal cells in the animal. I want those saved. Before I close, I just want you to remember that curing cancer sometimes isn't about fighting harder, it's about noticing the bigger nose. Thank you.